Hello students, today we are going to perform experiment 1, that is to prepare temporary mount of onion root tip to study different stages of mitosis. So here let us start the experiment. For this let us study what is required, what are the requirements for the experiment. So here you can see these are onion roots. Here you can see I will show with the help of forceps. These are onion roots. These are already fixed and dehydrated. Now another is here we need slide, here these are the cover slips we required, and here we need brush, here we need biological needles, forceps we required, here we need spirit lamp, here we can see spirit lamp, and here we need microscope. Here you can see the microscope, so compound microscope is with us, and here you can see the staining rack. And for staining we need acetocarmine. Here we need acetocarmine. Why acetocarmine is required? Because this stains chromosomes. So we can study the behavior of chromosomes. As you know that during mitosis chromosomes can be easily studied. Specifically in metaphase stage we can study the structure of chromosomes. So here let us start with the experiment. So what is required? So here you can see these are the onion roots. So what is required? We need tips of the onion roots, only tips, because tip is the portion where meristematic tissues are present. So you cut it with the help of needle or with the help of forceps. Now here I remove the tip of the onion root, because here meristematic tissues are present, and you know the characteristic features of meristematic tissue, that here the constantly dividing cells are present. Now what is the next step? The next step is pour 2 drops of acetocarmine and then heat it. Heating should be done very gently, else if you heat it harsh, then your material may be burnt out. So just to avoid, you do continuous, this is what is discontinuous heating is required. It should be done very, you can say, not the harsh. You just take your slide just above the spirit lamp, just to avoid burning of your material, else it will going to convert into ash. Now just to make it soft, now second round of staining with the same chemical, same stain, only 2 drops, that is acetocarmine, and then again discontinuous heating is required. And acetocarmine is highly volatile, so within 2 minutes it goes off, and you can see your material becomes soft. Now students, here you put the cover slip. Here you can see this is the cover slip. So staining should be done by same. Here you can see you have done staining with the acetocarmine. Even mounting is also done by acetocarmine. Now students, put the cover slip in this way and then tap it. Tapping should be done by blunt end of the pencil. So here you can first of all press it. Now here you can see the tissues become very soft. Now tap it. Tapping is done very precisely so that each and every cell gets separated from each other, and you can easily peep out within each cell that what cell division is going on. And then after that, excess of stain is to be removed by the help of blotting paper. Here is the blotting paper. So by the help of blotting paper, remove excess of stain. And then here is the use of microscope. You place your slide under the microscope and you can study different stages of mitosis. So when we study different stages of mitosis, then we can find that cells are at different stages. You can find prophase, you can find metaphase, anaphase and telophase. So this is what about your experiment. Now what to write? So what to write in your practical file. So here you write section A. This is experiment 1. What is the aim? To prepare a temporary mount of onion root tip to study different stages of mitosis. Now, what are the materials required? Here we required onion bulbs, though I have given you fixed and hydrolyzed onion root tips, so that is available to you when you perform the experiment. Beaker is required, fatty tissues are required, scissors, forceps, needles, spirit lamp, microscope, slides, cover slip, acetocarmine for staining chromosomes. Now here, how to fix, that is not required because we already give you the fixed and hydrolyzed onion root tips. Now observation, under the low power microscope you can see pink color nucleus, but under high power microscope you can see different stages of mitosis. Here you can see prophase, metaphase, anaphase and telophase. All the stages can be visible to you. Now here you can see this is what is seen under the microscope. Under high power microscope you can see different stages of mitosis. Here you can see this is prophase. This is the prophase stage. This one is the metaphase stage where chromosomes align themselves at the equatorial plate. Here you can see anaphase phase where the chromosomes get separated from each other, and here you can find telophase where within a single cell you can find two nuclei. So in this way you can see different stages of mitosis. So now come to next, here what to write about all the different stages. So these are given already. I'll share you this slide to the WhatsApp group, so you can note it down from there only. Okay, so here you can see prophase where chromosomes get condensed. Here you can see metaphase stage. These are the metaphase stage. This is early metaphase, this is late metaphase. You can see chromosomes align themselves at the equatorial plate. Here you can see chromosomes get separated from each other. Okay, longitudinal, before that longitudinal splitting of chromosomes resulted, and each chromosome has only one chromatid and they move apart from each other by the help of spindle fibers. And here you can see in telophase nuclear membrane reappears, nucleolus reappears, and within a single cell you can find two nuclei. So in this way you can see the result. So result is that you have seen all the four stages. What are they? Prophase, metaphase, anaphase and telophase. Now these are the precautions. Staining with acetocarmine should be done twice, and after each staining you must heat very gently so that material could not be burned out. So next is, the slide should be warmed gently, much above the flame of the spirit lamp. Tapping of the material should be done properly, precisely, so that you can get cells separated, and from each cell you can easily study the different stages of mitosis. Now while mounting, avoid air bubbles to enter. So this is what about your practical, and thank you and have a nice day. Thank you.